Thanks, Tim, very much. A couple of editorial comments. My primary area of research interest is cancer biology and autoimmune diseases. But however, I was trained as a viral immunologist, and I've always been working in the area of viral immunology. So when the economic disaster hit the country, there was ARA funding available, and NIH, in their infinite wisdom, they decided to give me about $1 million to spend in 2 years. And they said, you can do whatever you want. The primary goal of the budget is to do innovative research and also keep people employed in your lab. I said, terrific. So my student who was sitting here is gone to fetch a pointer, and his name is Hatem, and he very successfully defended his PhD thesis, and he's doing a postdoctoral work. So he and I, his background is pharmacology. So we decided, we are going to launch into a completely different area of research in my lab, and the basis for that thought was, there is what we call broad spectrum antibacterial, drugs that are available that we use against, treating many different bacteria. And then I asked myself, how come when the virologists try to develop a drug, we really go after the tiniest aspect of the viral life cycle, and we specifically target certain nucleotides or certain enzymes, that would actually negate the ability of the virus to replicate. And then we said, look, we have this money, and really there is not a whole lot NIH is expecting from us other than they want us to be innovative. So why don't we think of developing broad spectrum antiviral drugs? So that's what got me into Ebola, SARS, Nipah, Hydra, all these incredibly nasty viral infections. So what I will do is I will talk about our research primarily conducted by Hatem, he's here, and as the third part of my talk. The first part of my talk, since there is a very mixed group of audience here, I will spend about 15 minutes or so talking in general about Ebola virus. What is it? Why is it infecting people? And where is it affecting people, and so on and so forth. And part two, I will talk about what some of the protective measures that we can take, what is the risk for any of us in this room, and so on and so forth. And third part, as I already mentioned, I'll tell you a little bit about our own research. And so when I'm trying to please a diverse audience like this, I'm afraid I might displease everybody because there will be a little bit for everyone to pick on and a little bit for everyone to take home. So with that long introduction, let me start off by simply telling you what Ebola is. Ebola, also known as Ebola hemorrhagic fever, is a rare and deadly disease caused by infection with Ebola virus. And the word rare is becoming questionable, as we all know, based on what's going on in the news media. Ebola virus was discovered in 1976 near the Ebola River in what is now known as the Democratic Republic of Congo. Since then, the outbreaks have appeared sporadically in several African countries. The 5 Ebola virus subtypes that are closely related to Marburg virus, there is another nasty virus, and they belong to the family Filoviridae and order Mononegavirales. Mono is a single strand, nega means negative virus, and that's the name of the order. There are 4 types of Ebola viruses that can cause human infections, and they are Zaire, Sudan, Bundi, Bugyo, and Thai forest. As you can see, the death rates or the case fatality rate for Zaire virus is 79%, give or take, and Sudan is about 53%, Bundi, Bugyo is about 27%, and we haven't seen any human deaths due to Thai forest virus infection. The 5th virus is called the Reston virus. It has caused disease in non-human primates, but as of now, we have seen no human cases resulting from infection with this virus. Zaire Ebola virus is the most dangerous of the known Ebola disease-causing viruses, as well as being responsible for the largest outbreaks in humans. This is how the virus looks under an electron microscope. They are filamentous particles with negative single-stranded RNA as the genetic material, and that may appear coiled, toroid, or branched. The virus particles are approximately 80 nanometers in width, but vary in length, with median length ranging from 974 to about 1100 nanometers. You cannot see this in light microscope. It can only be visualized under electron microscope. And this is one of the very important definitive diagnostic criteria for Ebola virus infection. So this is just a map of as of August 25, 2014. This is how the cases looked. This is from WHO. I already told you the first outbreak was in 1976 in Congo, Democratic Republic of Congo. And this is where the current outbreaks are that are shown in New Guinea, Liberia, and Sierra Leone. That's pointed out here. And of course, this is in Zaire, where it started. And of course, we also have it in Nigeria and other parts of Africa that's shown in this map. And the interesting thing here, in the 1976, what we call epidemic, we had this many cases. The number of people survived are shown in blue, and the number of deaths are shown here. Roughly 2/3 of the people died from this infection. And then kind of it disappeared for a while, almost disappeared for a while, and then kept coming back up and down throughout the years. And in 2014, it has. This peak is by no means complete. This is as of that, but this number is going up quite a bit. And it's primarily, and I'll tell you why there is a massive upswing in this. So this is in Western Africa, where a lot of the focus has been. It's in Guinea, Sierra Leone, Liberia, and so on and so forth. And you have been hearing quite a bit about the troubles in this country and what has happened in terms of inability to implement public health measures. Because when they put them all in hospital, some few people decided that it's not a good idea, and they let everybody in the hospital go. And I'll tell you why it becomes very important, because contact tracing is an extraordinarily important step in stopping the virus spread, and that's what is being affected. I'm sorry. Another point I want to make this shows too. Another point I want to make in this graph is, even in 2014, look what happened in the month of March and April and May. It was kind of chugging along like it always has. But since that time, it has really shot up. And these curves are by no means, this is as of August 29th, and it has gone up further since that time. So there has been a very acute and very fast spread of this viral infection in certain parts of Africa. And look at this, there has been over 1,200 cases, and again, this has gone up in number. This is as of July 27th. And there were 427 cases in Guinea, and 319 of them died. So roughly 70%, 75% mortality. Sierra Leone, 525 cases, and roughly 50% of them died, about 40% of them died. And in Liberia, 249 cases and over 50% death. So on an average, Ebola virus infection, the case fatality rate is about 55% of the individuals who acquire the infection go on to die. In some instances, it can be as high as 95%. So what do we know as of August 28? So far, there has been suspected confirmed cases of over 3,000, and there has been roughly 50% of those patients, more than 50% of them have died. And out of these 3,069 suspected cases, 1,752 have been confirmed through laboratory testing. And I will tell you what they are as I move along. And these are the numbers for individual countries that I don't want to belabor this point, but the point is that there are quite a few cases in each of these countries, and the mortality rate, for example, here it's nearly 60%, here it's 65%, here it's about 50%, so on and so forth. So it's spreading faster than anybody like it to, and it's killing a lot of people. So how does this compare to some of the other hot button viral infections that we have been reading in the newspaper? And the Ebola is shown here, and this is the Marburg virus I referred to very briefly, but the first outbreak was in 1967. And SARS, all of us are familiar with. This is severe acute respiratory syndrome that started in China and spread all over the world pretty quickly, pretty scary. And this we have been reading quite a bit over the last 8 to 10 months. This is called Middle Eastern Respiratory Syncytial Syndrome virus. And this has been largely isolated in Saudi Arabia, but it has gone on to neighboring countries, and it is carried by camels, believe it or not. So here, when they emerged, the Ebola emerged in 1976, Marburg in 1967, and MERS, I told you, 2012, 2013, and SARS, was between 2002 and 2003. And since that time, this has not really resurfaced. And the last outbreak of Marburg was in 2005. So originally, a lot of these Marburg and Ebola, the outbreaks have been in Central Europe as well as in Africa. Of course, Ebola is primarily in Africa. And MERS has been primarily in Arabian Peninsula, whereas SARS was in China. And the suspected sources, look at the culprits in every one of these cases. These are fruit bats by way of monkeys and other animals. Again, fruit bats, it carries Marburg virus. MERS is also carried by bats, but it's transmitted to humans via camels. SARS is also carried by bats, but it's transmitted through civet cats that's sold in wet markets in China. So in every one of these nasty surprise infections that has affected the human species, the virus is being carried by bats. It's very interesting. And they all belong to different, they're listed here. These two belong to filovirus, these two belong to a virus known as coronaviruses. And the type of illness is hemorrhagic fever by both Ebola and Marburg, whereas acute respiratory syndrome caused by both MERS and SARS. So they affect different parts of the body. And the fatality rate, I already told you, is 50 to 90%, depending upon the particular epidemic. And whereas Marburg is about 24 to 88%, and MERS was approximately 30%, whereas SARS, by comparison, you can see SARS is a sort of not such a bad disease, but nobody wants to get SARS, and let alone Ebola or Marburg. And I think the known cases are there, known deaths have already And then the transmission is person to person, readily by close contact or body fluids, not by casual contact. This is again readily by close contact or fluids, not by aerosol. Not very readily, and the transmission is unknown. And of course, the SARS virus is predominantly transmitted by aerosol, but also body fluids and close contact can get you infected. So this just to give you a sense of some of the hottest viral infections that we have been reading in the newspaper and as well in the scientific literature, and how does Ebola compare to those other three nasty viral infections. Ebola by far is the champion in destroying human lives and animal lives. So how do we diagnose Ebola virus infection? Well, the most important, believe it or not, is medical history. And especially you should ask them a lot of questions about the travel and the work history, along with exposure to wildlife are important in order to diagnose Ebola virus infection. Of course, these will all be suspected cases, as you can imagine. And during an outbreak, virus isolation is often not feasible, because if you are working in the field and you need a BSL-4 facility, wearing a space suit to work in the lab, it's not going to be feasible. Therefore, the most common diagnostic methods are we use real-time polymerase chain reaction. Basically, it's a way to amplify the genetic material and then look at it and see whether it is Ebola virus or not. And of course, we also can test for by ELISA. It is nothing but a serology test. This is to detect viral proteins. And one can also detect antibodies in patient serum. It is quite helpful, but it is in a very late phase of the disease, so it's not going to be of much help in terms of initiating treatment for the patient. But it will be very important to finally confirm the suspected cases as being really Ebola virus infection. So they all serve incredibly important clinical purpose or treatment purpose or public health purpose, because we want to have an accurate count of how people were infected, how many were infected, who got better, who didn't get better, because it will help us design interventional strategies. Ebola viruses can be seen and identified in cell culture by electron microscopy due to their unique filamentous shapes. But electron microscopy cannot differentiate between various filoviruses. They all look the same under the microscope. So you need a combination of serology, PCR, as well as morphology, along with case history, is how you're going to be able to diagnose Ebola virus infection. So how does this disease progress? Well, symptoms usually appear about 8 to 10 days after exposure. This is really a sort of an average, but the symptoms have appeared as early as 2 to 3 days after infection. So that's what makes it even more challenging. At first, people develop flu-like symptoms. They complain of headache, and of course they develop fever, and they are going to complain a lot of aches and pains throughout the body. And sometimes rash appears followed by diarrhea and vomiting. In half of the cases, Ebola takes a severe turn, causing victims to hemorrhage. Vomiting blood or blood in urine or bleeding under the skin, eyes or mouth are other hallmarks. Those are the hallmarks of advanced clinical disease. Bleeding, believe it or not, bleeding is not usually the cause of death, although everybody thinks, oh my God, people are going to bleed and they're going to bleed to death. They don't. In fact, the increased permeability of the deep blood vessels results in loss of body fluid, and there is electrolyte imbalance. That's what actually kills the patient. It's not the hemorrhage itself. Blood pressure drops, and of course that can result in failure of the heart, kidneys, liver, and organs as a consequence of loss of body fluid. So everything, cascades. A lot of attention initially was paid to hemorrhage, but that's not really it. And so these are the symptoms. I already told you they have headache. They develop sort of because of the increased vascular permeability. The eyes look very red. Systemically, they develop fever. They lose appetite, and there is internal bleeding. I already told you there is lots of aches and pains in joints and muscles, and diarrhea, skin rash and bleeding, vomiting, and you can see there is essentially the whole multiple organ systems are affected. And through in the respiratory system, there is hiccups, there is sore throat, difficulty in breathing, difficulty in swallowing, all due to the clinical signs and symptoms Ebola evokes in infected individuals. These are actual pictures of actual patients. As you can see, the clinical picture is not very pretty once they get into advanced stage of the disease. And so this is pictorially, I'm just showing you what I already told you. This is just to, so that one can have this in a field hospital so that people know what to look for. It's a constant reminder. This is put out by CDC. First symptoms, 7 to 9 days. I already told you headache, fatigue, fever, muscle soreness. And of course, there is a spiking of the fever, vomiting, and passive behavior, as well as passing of the blood in the stool. And then you will have brain hemorrhage, bleeding from nose, mouth, eyes, and anus that I showed you some pictures of. And the final stage is loss of consciousness, seizure, massive internal bleeding, and death due to multiple organ failure. And this is what, given where this disease is predominant, so these kinds of signs are used in all the affected areas, so that if people can recognize the signs and symptoms of the disease and immediately seek medical care. And this is obviously this is what the public health people are doing and the doctors are doing, and then there is a whole cultural milieu in which these things are happening, and sometimes these become incredibly ineffective because the belief systems dominate over logical thinking and all the good intentions people have. So where did the disease come from, or where does the disease come from? I already told you it was discovered in '76. And one started to originate in gorillas because human outbreaks began after people ate gorilla meat. Most of us refer to it as bush meat when they eat the non-human primate meat, and everybody thought that's how they got infected. But that theory has been ruled out, partly because apes that become infected are even more likely to die than humans. Humans, I told you, can live up to 12, 14, 17 days after infection, whereas most of the non-human primates die within 5 to 7 days. And therefore, it's unlikely, people got the infection because of that. It is now believed that the bats are the natural reservoirs, and the apes and humans get infected by eating food that bats have drooled or defecated on, or contact with surfaces contaminated by bat droppings. Look at the way we are acquiring this infection. And this is terribly, terribly important because how we think the infection spreads is going to decide on what intervention strategies we are going to develop. So therefore, understanding this was incredibly important. The current outbreak seems to have started in a village near Gokedo. It's in Guinea, where bat hunting is very common. Okay, this is from Doctors Without Borders observation. A lot of these are observational type of studies rather than solid scientific controlled studies. So how is this virus spread? The virus spreads through direct contact, through broken skin or mucous membranes, blood and body fluids, urine, feces, saliva, vomit, and semen of a person who is sick with Ebola obviously becomes the source of the infected material. Objects like needles that have been contaminated with the virus. Ebola is not spread through the air or by water or in general by food, but of course the contaminated food can give it to you. However, in Africa, Ebola may be spread as a result of handling bushmeat, wild animals hunted for food, and contact with infected bats, which we know is the primary cause of acquiring infection from wild animals. So this is basically a simplistic diagram of Ebola virus transmission and spread. As you can see, the virus can go from bat into animals, and then the bat flies around and it gets into all kinds of situations, and eventually the humans end up getting infected primarily because of infected bats. But of course, sometimes eating bushmeat from a clinically not apparent infection of non-human primates. This is probably the most important slide I'm going to show you, because this is what's going to help people decide on who may be infected, who may not be infected, and also how to contain the infection. So that is shown here. You have an Ebola patient who is undergoing patient care. And then this is how you triage these cases. You ask about the contacts and take all kinds of history that I already described. And if a person has been in contact with a patient, obviously this patient needs to be treated. And if this patient, then you have to further investigate and ask, who else was in contact with this person. And again, if there is a person who was in contact, you need to observe this person for a minimum of 21 days before you declare that the person is not infected. If there is any signs or symptoms, obviously immediately you subject them to care. And here, if a patient is known not to be, there are no symptoms, if the contact shows no symptoms after 21 days, obviously you can safely let them go home. And however, if you have a mixed contact, we don't know whether the person was in contact or not, but then therefore you still follow the patient for 21 days before, the subject for 21 days before you let them go home. And this goes on and on. So contact tracing finds new cases quickly, so they can be isolated to stop further spread. If this is really our central interventional strategy to spread the epidemic, to stop the epidemic, look what they did in Liberia. They let the infected people get out of the hospital. God knows how many more people they have come in contact with. And so therefore, the very tool that we have to stop the spread of the infection has been taken away from us, number one. Number two, earlier, the incidence of these cases were very few because it was mostly in highly isolated rural communities. Now it has gotten into urban communities. And therefore, tracing the contact and containing them and treating them and monitoring them has become a challenge because contact tracing is not that easy. So that's what is also causing this massive spike in the number of cases. And the CDC director just came back from a big trip in Africa. He's very concerned. He's incredibly concerned, and he's been on every news television network this morning urging the governments to come together to deal with it. And the Guardian newspaper in England has an editorial urging the government to come together to put boots on the ground to contain the infection, because this has gotten into urban areas, and now it can spread quite rapidly. And the second thing that has happened is a lot of public health workers from abroad, the governments and the nonprofit, the NGOs, they are not allowing people to go and work in this area because of the increased risk as well as threat to their life. So everything that can go wrong that we think of in some sort of an epidemic is going wrong here. We don't have a treatment. We don't have proper government structure in countries where this disease is rampant. And number three, the healthcare workers internationally and nationally are not available to deal with them, and just going on and on and on. I don't mean to scare you, but that's the reality that we are facing, and as spoken to this morning by the CDC director.
Rob,
Yeah.
just a quick question. So if quarantining for 21 days is essential, what are the means they have to quarantine? You say they let them go out of the hospital, but a lot of these areas don't have proper quarantining.
So they're
facilities.
quarantining, essentially, you use PPE, personal protective equipment of the healthcare. So it's basically, you can't keep the patient in isolation. The doctors are isolating themselves from the patient. And through personal PPE, it's called personal protection equipment. And I'll show you pictures of that. That's obviously a relevant question. So who is most at risk of getting Ebola? Healthcare providers, you can see who is going first. Caring for Ebola patients and family and friends in close contact with Ebola patients are at the highest risk of getting sick. And the very people who are trying to save lives, they are putting their own life at risk. They may come in direct contact with the blood or body fluids of sick patients. In some places affected by the current outbreak, care may be provided in clinics with limited resources. That's the understatement of the year. Workers could be in those areas for several hours with a number of Ebola-infected patients. So the longer you are exposed, the higher the probability of acquiring infection. Additionally, certain job responsibilities and tasks, such as attending to dead bodies, may also require different PPE, that is personal protection equipment, than what is used when providing care for infected patients in a hospital. Because in the hospital, basically you're giving supportive therapy that I will get into, whereas when you're dealing with cremation the level of contact and the extent of contact is going to be quite different. So this is what they're doing, Tim. You can see this, but this is before you go see a patient, you don your spacesuit and then you go, care for the patient. And look what is happening. These are all the clothes and gloves that had already been used for patient care. This is how they are sterilizing and reusing it. Yeah. Just to show you the complexity of the problem, this is in a field hospital. So not likely to, how contagious is the virus? You're not likely to catch Ebola by being in proximity with someone who is infected. Ebola transmission is not airborne, like the flu or respiratory virus such as SARS. Ebola spreads through direct contact with body fluids. If an infected person's blood or vomit gets in another person's eyes, nose, or mouth, the infection may be transmitted. In the current outbreak, most new cases are occurring among people who have been taking care of the sick relatives or who have prepared an infected body for burial. Healthcare workers are at very high risk. If they have not been properly equipped with or trained to use and decontaminate PPE correctly, they are going to be obviously putting themselves at higher risk. The virus can survive on surfaces, so objects contaminated with body fluids like latex glove or a hypodermic needle may spread the disease. And again, it gets back to bad droppings and how it can contaminate the surfaces and so on and so forth. Okay, so here is, Tim, another picture to answer your question. Here is a patient in a field hospital, and look who is being protected here. The care provider is the one protecting himself from the patient, because as you rightfully said, there are multiple patients in a big room, and there are not isolation wards. Nobody is controlling the airflow or HEPA filters or all the usual stuff that we think of. And this is how a CDC bed looks. See what they have done is they are completely isolated, and the care provider is outside this latex tent, and that is the bed on which a patient in CDC or Emory would have slept. Yeah? So look at the difference between what we are doing here and what's going on in the field. And this is the isolator that was used. It was retrofitted into an aeroplane. These are aeroplane seats. This is how the Spanish priest was transported from Africa back to Spain for the treatment. So this is how the isolation chamber was created. To give you a sense of the challenges, both technical and medical, that one is facing. So just to put all of you at ease, what are the chances of Ebola in the United States? 2 American aid workers infected with Ebola virus while working in West Africa were treated at Emory Hospital in Atlanta. Both were released a couple of weeks ago, and they've gone back, and their children and family have gotten together and have given television interviews. So that's very good news for 2 of our own. Doctors across the country are being reminded to ask for, because you don't know who is coming, because we have a lot of people traveling to Africa and coming back, the travel history of anybody who comes in with a fever. This is what we are supposed to do. Patients who have been to West Africa are being screened and tested if there seems to be a chance they have been exposed. False alarms raised at 3 hospitals in New York City, but no Ebola cases have turned up. See, this is the worst thing that happens. Whenever you have a false alarm, people go through that false alarm once or twice or thrice, and then they say, it's a false alarm. And they don't, they drop the guard. And this is really one of the nastiest things that happens in public health. If someone were to bring the virus to the US, standard procedures for infection control are likely to contain it. Ebola does not spread nearly as easily as Hollywood movies depict, and I'm sure all of you have seen Dustin Hoffman and others scare us. So why is Ebola so difficult to contain? Well, the epidemic is growing faster than efforts to keep up with it, and I already alluded to some of the problems. It will take months before governments and health workers in the region can get the upper hand. This is according to Doctors Without Borders. In some parts of West Africa, there is a belief that simply saying Ebola aloud makes the disease appear. I mean, this is a fact. This is a fact, as incredulous as it sounds. Such beliefs have created major obstacles for physicians. Some people have even blamed physicians for the spread of the virus. Believe it or not, this is not as dumb as it sounds. Their skepticism is not without merit. Why? Because in the past outbreaks, hospital staff members who did not take thorough precautions transmitted the virus. Imagine what's going on here. And you're the person they're trusting their life with, and they acquire infection from you, and the word gets around. Ball game is pretty much over by that time. So how is the disease treated? Well, the most important slide probably in terms of for patient care is here. No Ebola virus vaccine or specific antiviral drug exists. So that's a big negative declarative statement, which is true. Treatment is primarily supportive in nature, and that includes minimizing invasive procedures because the chances of other people getting infected and everything goes through the roof. Balancing fluids and electrolytes to counter dehydration. Probably the most important thing you can do is that. And the second most important thing is to maintain blood pressure through medication. Yeah? Because of the electrolyte imbalance, cardiovascular system fails, and therefore blood pressure drops primarily. So you want to, stabilize the patient. And of course, some patients appear to have immunity and nobody knows why. And I can speculate. Probably the receptor has a mutation, therefore, the virus cannot get into the cell. But that's for the geneticist. Anticoagulants administered at early infection may prevent or control disseminated intravascular coagulation. Procoagulants administered late in infection may control bleeding. Maintaining oxygen levels and pain management obviously are important. Treatment of secondary bacterial fungal infections, because believe it or not, this is the third most important thing you can do. Early treatment may increase the chance of survival, as has been shown. And I read another story this morning. A patient in Great Britain in a hospital in London was released today, and he was admitted about 10, 11 days ago with Ebola infection.
Can I ask a question? There were reports that these two patients who were brought to America got some serum. Okay.
I'll get into that. So, because I have to build that up to tell my story.
So earlier the graph showed in Sierra Leone there was about 50% survival or 55% mortality, but the other ones were less than 50%.
Yeah.
So is it because there's different or better or less equipped facilities, or because why is the death mortality different?
It's a combination of multiple factors. Typically, there is varying genetic susceptibility for all infections. And if all of us in this room got exposed to, we were riding a subway and we all got exposed to the same sneeze, and maybe 3 of us will come down with the flu, but most of us will get the infection. So I think there is a different level of immunity, different genetic predisposing factors, so on and so forth. And of course, in this case, most importantly, maybe the differences in how early the treatment was initiated and how effectively the patient, treatment was administered. I think multiple variables. But right now they are so busy, treating and containing the infection. These kinds of information will come out when they go back and do retrospective analysis. That's an important question, though. So how do I protect myself Ebola? If you are traveling to an area affected by Ebola outbreak, protect yourself by doing the following. Wash hands frequently. Avoid contact with blood and body fluids of any person, particularly someone who is sick. Do not handle items that may have come in contact with an infected person's blood or body fluids. Do not touch the body of someone who has died from Ebola. Do not touch bats and non-human primates or their blood and fluids, and do not touch or eat raw meat prepared from these animals, of course. Avoid hospitals where Ebola patients are being treated. Look at that. If you're going there to help, they're telling you to avoid. I mean, this is really how crazy all of this becomes. Believe me, I was on a team at CDC for 3 months. I was hunkered in the basement at CDC to develop a pandemic flu preparedness program. So I was one of the lab scientists that was selected for that. I mean, these are the kinds of things that we dealt with almost every day, because when all hell breaks, all hell breaks. And seek medical care immediately if you develop the fever and any of the other following symptoms. Headache, I already went through that. Limit your contact with other people until and when you go to the doctor. Do not travel anywhere else besides a healthcare facility. U.S. citizens should avoid all non-essential travel to all these countries. Travelers to Nigeria should take enhanced precaution to prevent Ebola. So there are multiple levels. There is level 3 guidance and there are level 2 guidance. So level 2 you can go, but be careful. When level 3, avoid unnecessary travel to those parts of the world. Okay, yeah.
So even though it's transmitted by bodily fluids, as you said, if you come into contact with things, like you showed that picture of the gloves.
Yeah.
So I've heard that the virus can live on other
surfaces for a
objects.
long time.
And would bleach destroy it? Because that's commonly the case in most places where
Yeah.
like they reuse gloves over
Yeah,
and over, and it's not that easy
Yeah.
to just dispose of
Yeah,
them.
yeah.
But if-
So the bleach also eats into this latex material. So in the laboratory we use Vescodine. It's an iodine-based, there are many different things that you can use that are non-corrosive. But the problem is, they're dealing with this crisis situation. They themselves are scared. It's just, I think all of these things.
If you don't have enough personal protective equipment to just dispose of it every time.
That's what-
It can
Yeah.
remain on the.
Absolutely. Well, but not if they were properly, disinfected prior to, washing and reusing.
Prior to reusing.
Absolutely. So these are the experimental therapies that somebody has. So what do we do typically for a viral infection? The antibiotics work only against bacteria, correct? So we don't have an antivirals against all imaginary viral infections. We have only against a handful of viruses. So therefore, most of the viral infections are prevented because of active immunization. As children, when you go to the pediatrician, they keep giving you shots, correct? If you think about it, most of them are for viral infections, mumps, measles, polio, on and on and on. So, then therefore the central question is, are there vaccines available? The answer is no. But what are they doing? There are currently no FDA approved vaccines for Ebola. NIH is working on developing an Ebola vaccine, and these are some of the efforts that are underway. NIH will launch phase 1 clinical trials of NIAID, GSK NIAID is the Allergy Infectious Disease Institute at NIH. GSK is GlaxoSmithKline, Ebola vaccine, and they can confer protection against both Zaire and Sudan strains in chimpanzees, okay? And the clinical trial will start this fall. NIH is supporting Crucial Biopharma Company, and again, it's a combination vaccine against Ebola and Marburg. NIH is supporting Profectus Biosciences, and they're preparing an Ebola vaccine. NIH and the Thomas Jefferson in Philadelphia are collaborating to develop a candidate Ebola vaccine based on the established rabies vaccine. So look at the treatments of choice here. NIH, DoD, and New Link Genetics Corporation, which I think is a Canadian company, and they're creating a VSV Ebola vaccine, and that's going to be tested in Walter Reed Medical Center. So these are some of the most current efforts that are underway to develop a vaccine. As you can see, every one of these vaccines is a subunit vaccine. Obviously, you're not going to use the whole virus like we do for measles, mumps, and polio, correct? Because you don't know when it can revert back into being an incredibly pathogenic strain. So we take pieces of Ebola virus, and I will show you a picture of that. And we basically use the vesicular stomatitis virus as a backbone, and we patch the viral protein onto it, and then it's a VSV virus with Ebola protein, and therefore we make an immune response to that. And that's the same thing with many of the other viruses, other vaccines. So passive antibody protection, this is the one that you're talking about. And there is a product known as ZMAP, and this is produced by a company called MAP Biopharmaceutical. It's in San Diego, California. Basically, it's a combination of 3 monoclonal antibodies. You talk about technology convergence. These antibodies are produced in tobacco leaves, believe it or not, and then they are purified. So it's a cocktail of 3 antibodies. And 2 of the US and 1 British patient, as of this morning, have been treated with this antibody, and all 3 have survived. However, a Liberian doctor and a Spanish priest who were also treated with this antibody, obviously they were not as lucky. Both of them passed away. So that leaves us, with a very confusing situation. Did these antibodies really help, or it's basically, supportive therapy that got these people, Because the supportive therapy here was given at Emory Hospital, and in Britain it was given in one of the leading hospitals in London. I forget the name. So I don't know whether it is the quality of the supportive care or the combination of supportive care and passive therapy. We don't know. And the most exciting news, as of, believe it or not, I think as of last Friday, the report came out, 18 rhesus monkeys were treated with ZMAP, monoclonal antibody cocktail, and 5 of these non-human chimpanzees were treated, rhesus monkeys were treated 5 days after the infection took hold, and they were showing clinical signs and symptoms of the disease. And yet, 100% of the 18 monkeys that were treated at different times after infection have survived. This is probably the best news I can give you this afternoon.
And what is the name of the product once more?
That is ZMAP.
ZMAP.
Yeah, so now we have very direct evidence that ZMAP can actually protect, not only given prophylactically, but given therapeutically, even when the clinical signs and symptoms. So therefore, now the recommendation is if the patients are 9 to 11 days into the infection, still this antibody could have some protective effect. So that's the current belief. It's a speculation. And of course, the next thing that one can do to provide instantaneous protection if you do not have vaccines available is antiviral small molecules like we do for many viral infections such as HSV, HIV, and so on and so forth. So I will take another 10-15 minutes and quickly walk you through. I'm not going to bore you with all the technical stuff here. It's a complex viral. This is how it looks, filamentous. This is the glycoprotein that's probably most important for eliciting a protective immune response. And this is simply compared to other, this is Ebola virus genome, this is the Marburg, and in general how the structure looks. They've got a whole bunch of different proteins. And the reason this virus is very nasty, for example, VP24 and VP40 have been clearly demonstrated to suppress innate immune responses. So when a cell is infected, we have evolved to mount a very brisk, what we call innate immune response in the form of interferon production. And these interferons then act on many different transcription factors by binding to interferon receptors, and they kick up the cellular defensive machinery. And that is through a signaling pathway known as STAT4. What this does is these guys bind to the very signaling molecule that is necessary to go into the nucleus and turn on the host cell defenses in response to interferon. And therefore, the interferon-mediated protective effect is completely abolished. So these viruses are not only nasty in terms of causing clinical signs and symptoms, they have also evolved to evade the host immune system so that they can persist and cause disease and can be transmitted to other individuals. So again, I'm not going to really bore you with all, no, let's skip this, it's too much virology. Okay. So how do viruses get into a cell? Well, for enveloped viruses, basically it has to fuse with the host cell membrane. And those membranes can be either plasma membrane or endosomal vesicle membrane that is surrounding the endosomal vesicle. And that's what Ebola is. And for non-enveloped viruses, such as, for example, polio, they form a pore and other means of permeabilizing the lipid bilayer and that they go straight into the cytosol, whereas this guy has to go through a fusion step. And I'll tell you why it is important. So this glycoprotein fuses with the endosomal membrane and then opens up the membrane, and then the viral genetic material is released. And here, for example, this is the polio where the material is coming without fusion. Whereas here, you can see they come in, they're now enclosed in endosomal vesicle. Upon fusion, the endosomal vesicle opens, and then the virus particle is released, and then the machinery gets turned on, and you have fresh viral particles being generated. So this fusion step is what is very, very, very interesting about the Ebola virus. And so this is how the Ebola virus enters into a target cell. So there is a receptor. The first receptor encoded is known as Niemann-Pick C1, NPC1, and there is other proteins called DC-SIN, there is STIM1, and so on and so forth. And basically, they bind to a number of different cell surface proteins, and they enter into the endosomal vesicle, and then there is an enzyme called cathepsin L and cathepsin B, and they basically cleave the glycoprotein and allows the glycoprotein to fuse with the membrane, and therefore the virus particle can escape from the endosomal vesicle and start replicating itself before it can get out of the cell. So that fusion event is incredibly important, and that's again shown here in a more dramatic way. And once the virus gets out of the cell, it can infect monocytes, and you can see what the monocyte infection results in. It ends up pumping out tremendous amount of pro-inflammatory cytokines like TNF-alpha, interleukin-6, interleukin-8, and also can cause cell death due to cytokine storm. It is the cytokine storm that actually causes much of the hemorrhage, number one. Number two, it down-regulates integrin expression by viral glycoprotein shuts down the integrin expression. So integrins are the ones that keep what we call tight junctions, correct? Now the integrin is not there, the cells start falling apart. That's when you start losing body fluid, okay? And if it happens in the vascular endothelial cells, you're going to have bleeding. So that's what is causing. So disruption of the cell-to-cell contact, integrity of the cell-to-cell contact leads to two disasters. One is electrolyte imbalance and acute body fluid loss, which causes systemic failure. And also, if it happens in vascular endothelial cells in deep vessels, you're also going to get hemorrhage. So that's the reason why Ebola virus causes the signs and symptoms it does. So remember, I told you we were very much interested in developing a broad spectrum antiviral. So here is a SARS virus, here is Ebola virus, and here are two other very nasty viruses that are not in news yet, but I can assure you they will show up one of these days, called Hendra and Nipah, because they are like Marburg, Ebola, they're all like that, in African jungles. And they're envelope proteins, and they're all class 1 fusion proteins, and they require priming through cleavage with host proteases. Look at what they do. They get into the cell, they exploit a host enzyme called cathepsin L to cleave the glycoprotein and cause the fusion and then release the virus. If there was no cathepsin L, we won't have Marburg virus infection, most probably, okay? Unless it gets adapted to some other. So cathepsin L is nothing but a lysosomal host protease. It is required for priming the glycoprotein so that it can fuse with the membrane and release the viral content. So I'm just going to go through all of this quickly. So you may say, why don't you target cathepsin L? Well, the problem of targeting cathepsin L, many people have done this. But the limitation of that is cathepsin L is an important host protease. It is involved in the processing of neuropeptides. So you may cure patient of Ebola, but you may also kill the patient because if you look at all the important neuropeptides that cathepsin L is involved in processing. Remember, most of the neuropeptides are formed as pre-propeptides or propeptides, and the active form results only upon cleavage by cathepsin L or enzymes like that. So cathepsin L is involved in releasing enkephalin, neuropeptide, as well as dynorphin, cannulin, so on and so forth. So you cannot shut down the function of cathepsin L and keep the host alive. You may cure the patient, but kill the patient at the same time. So what did Hatem actually do? Okay, so we did something very cute, I think. So what we did was we synthesized the peptide that contains the cathepsin L cleavage sites from glycoproteins of all 4 viruses that I told you: Nipah, Hendra, Ebola, and SARS, because they all use membrane fusion to get in, and they all depend on cathepsin L to get into the cell, okay? So they all have something in common. So we synthesized a peptide. At one end of the peptide, we put a quencher, and they quench light. It's called TAMRA. And at the other end, we put a light emitter called 5-FAM. Because the peptide is short distance from one end to another, the light emitted by 5-FAM is quenched by TAMRA, so you get no light at all. So when you put it in a luminometer, you get no light. But however, if the peptide is cleaved by cathepsin L, now you're separating quencher away from the light emitter, and there comes the light, okay? So the amount of light you generate is directly proportional to the degree of cleavage that can create in the peptide. So now we use this as a screening method, and I said, okay, if I have a drug that can prevent cathepsin L mediated cleavage of this peptide, then there should be no light, correct? In the absence of an inhibitor, the enzyme should act and it should release light. That's a very simple high throughput screening assay. Thanks to Hatem and his imagination. Nice to have a pharmacology expert in the lab. And this simply talks about the reproducibility of the result, and look at the fluorescent light being emitted because of the cleavage. And you can see, you can keep doing it hundreds of times and you get the same result. And this is when you don't have any enzyme. So I don't want to, and the most important thing I want to point out here, because this becomes terribly important. If you look at SARS, Ebola, Hendra, and Nipah peptides, and the cleavage rates are at 1 microgram per ml is around here, that's shown, this lower amount. But look what the enzyme has very high affinity for the host peptide. So this is what we have taken advantage of. So evolutionarily, this enzyme was meant to process our neuropeptides. And therefore, they have very high affinity for the host peptide or the host protein. And however, these viruses have evolved to take advantage of the host protease for their own gain. And therefore, by definition, we would have suspected that they would have lower affinity because it was not meant for viral infection. So we took advantage of this differential affinity, and we conducted high throughput screening. So basically, everything that you see in dark bold letters here, showing much higher protection of cleavage by the small molecule compound library compared to the host peptide. So wherever the host peptide was not protected, we can see it was still protecting the viral peptide cleavage. Yeah, that's what these numbers. So we went through this massive screening, and we identified a bunch of peptides. that showed there are about 12 of them listed here. They showed varying degrees of protection against Ebola virus cleavage. And the next one shows basically it had zero protective effect. These compounds had zero protective against another virus known as VSV, vesicular stomatitis virus. You can see there was 100% infectivity and there was no protection against VSV infection. And then also we wanted to make sure that these compounds were not cytotoxic, and that's what the cytotoxicity assay is at various doses. And again, I don't want to bore, except for this compound, all the other 4 compounds had little or no cytotoxicity, only that one was, so we discarded that because we don't want the drug to be killing you. And then we said, remember, we used only peptide for the screening. So we wanted to ask the next question, can these compounds really protect the enzyme from cleaving the entire protein? So we synthesized the full glycoprotein of the SARS virus here, and you can see that in a dose-dependent manner, the viral peptide can be very easily cleaved. Now, in the presence of this compound, you can see, the cleavage product, the cleavage is being shut off. Yeah? So higher the dose of the drug, the less the cleavage of the protein. That means, in the presence of this compound, theoretically, the SARS virus cannot infect the cell because the glycoprotein is not cleaved by cathepsin L, and therefore the virus cannot establish a shop inside the cell. And we have done this with a derivative of this compound. You can see that's even more dramatically protective against against that. And of course, there are controls here when we use cathepsin L inhibitor. Obviously, and this is with Hendra and Nipah virus, the same concept. And you can see we looked at the entry inhibition of pseudotype viruses where we deliberately take glycoprotein from the nasty virus and we put it onto an HIV backbone. But the entry depends on the viral glycoprotein and its ability to bind to the cognate receptor. So this is called pseudotyping. So when you do the glycoprotein from Nipah and Hendra, we get nearly 100% protection from viral entry into the cell. And also it prevents fusion. And you can see if the virus was infecting the cell pseudotype viruses, it will cause giant cell formation with multinuclei. The blue spots are all nuclei. And in the presence of the compound, you can see there is hardly any fusion, suggesting that we have a couple of lead compounds which are now capable of preventing cathepsin L-mediated cleavage of the glycoprotein from 4 different viruses. And they have selective inhibition of cleavage of viral glycoproteins, but not host proteins and all of that. So what then we wanted to know, there are, the inhibitors can be competitive, non-competitive, uncompetitive, or mixed. And I can tell you our inhibitor is a mixed one, and these are the enzyme kinetics, and I really don't want to bore you with all that. So in summary, we believe we have developed a novel assay to screen for broad-spectrum antiviral compounds. We identified a broad-spectrum antiviral small molecule against SARS coronavirus, Ebola, Hendra, and Nipah viruses. The small molecule is a mixed inhibitor of cathepsin L and can inhibit viral entry. These have to be obviously tested in BSL-4 facilities against live viruses, which is being done at NIH. They have actually taken our compound for further testing. Those small molecules with antiviral effects will have to be further developed and tested in non-human primates before it can be used in human beings. So with that, I'm going to end my talk, and I'll be happy to answer any questions. Obviously, much of the credit should go to Hadi.
Questions for Dr. Prabhakar?
Yeah,
Where did you do the titratable screening?
here at the College of Pharmacy.
Okay.
So in terms of the mechanism, when you block the cleavage, that means that you don't get the glycoprotein. The glycoprotein is not being released. That's what stops the.
It's not being processed.
It's not being processed.
Yeah. Because see, the glycoprotein, when you cleave it at that site, then it can fuse with the endosomal membrane and they open it up.
I see.
And that's when the genetic material gets into the cytosol. Otherwise, it just sits in the lysosome and never released into the, into the
So
cytosol.
cells will have
The cathepsin L. It can only infect cells that express cathepsin L.
so
And most of the cells, unfortunately, express cathepsin L.
yeah. So when you block it, so do you think that there will be some accumulation of non-cleaved material in cells, or whether that can be?
No, so whatever the number of viral particles that enter, remember, the virus has not undergone any replication.
Okay.
So therefore, there is no increase in overall glycoprotein load. Whatever got in is sitting in the lysosome. That's it. And also, there is most of the cells once they're infected, they become refractory to super infection. Those are all different viruses have different properties. Most of the viruses, when they infect a cell, that cell becomes refractory to further infection because they want to now take over the machinery for replication and not keep getting stuff from outside.
Sounds fantastic.
I think so.
Can I ask you, in terms of the natural course of the disease, in that first week when people are asymptomatic, are they still considered infectious? And for the, I guess now it's several hundreds of people who have survived infection, how long does it take until their body is officially free of virulent particles?
Well, after 21 days, they're released into public. So by definition, the viral infection is clear because they see no trace of viral infection, either by EM or by any other means. And unfortunately, most viral infections are most notorious during asymptomatic phase. I mean, if we knew, if I have cold, by the time I have cold and I got a flu and I go to my office, believe me, I'm not in danger to anyone.
Might that be different? I mean, if it's transmitted by vomiting and blood, and asymptomatic and doing neither.
Yeah, but prior to that, that's why the physicians have to be super careful. The moment you think it's a suspected contact, you're going to treat that person as a case. As a healthcare provider, you have no choice. That's why the case contacts are so important, and that's what was undermined by breaking into this hospital. Because now we don't know where these people have gone. Some of them gone back to villages, some have gone back to some other country. I mean, the whole thing is insane from a public health interventions point of view. Yeah.
So the drugs that you've identified with these assays, are they still presenting the problems of cleaving the neuropeptides also, or was this?
Well, the answer based on simply the assay that we have done, the amount of drug that we use completely retains the host peptide cleavage function while can inhibit the viral cleavage. Why is it? Because that's why it's a mixed inhibitor. So what happens is the affinity of the binding of this drug to, let us say, the peptide enzyme or the protein enzyme complex is such that only it can displace the enzyme peptide or enzyme protein interaction if it's a viral protein because it has a lower affinity for the enzyme or vice versa. But because the enzyme has such a high affinity for the host peptide, it can displace the drug and still attack the host pre-protein and process it. You understand? It's all relative affinity phenomenon that we are dealing with.
Other questions?
Yeah.
Yes.
You mentioned that when the director of the CDC went to different African countries that he said it was in really bad shape. What do you think, do you have any thoughts of what they should be doing right now at this point?
Really, the simple answer is get hold of the people who have ever been in contact with a patient or a suspected patient and quarantine them for 21 days. Really, the solution is not that difficult. But how do you find out who the contacts are and who the suspected cases are? That's the challenge right now. And because it has penetrated the urban area and all these beliefs or disbeliefs they have, that's causing a lot of the problem too. And people are hiding in the sense, if they have the symptoms, they don't want to go to the doctor because, they're living in somewhat in denial. It's very tough. Very tough. I mean, the simple solution is massive public health intervention. That's what's going to stop this. That's why I told you, the Guardian editorial says put the boots on the ground. I don't know which boot is on the ground is going to go there and deal with Ebola cases. I mean, I don't understand any of this. Therefore, what it requires is highly dedicated healthcare workers, but unfortunately, their own lives are in danger because of this chaos. I honestly believe in next week or two, there will be very clearly enunciated policies because for the first time, the CDC director was on the ground and has really assessed the situation and come back, and he's putting alarms all over the place. And therefore, the UN, WHO, everybody has to get in on this act and put our collective efforts together and see whether we can contain the case. I mean, Liberia is, Monrovia has been, I don't need to tell any of you what the mess over there. These are all failed states. I mean, that's the danger. Here, all of a sudden, the politics is getting mixed up with public health. And that's literally where these viruses, take off.
Right.
It's been the case forever. Yeah?
Just, is it MSF are the main people that are on the ground?
What is that?
Currently, MSF and
Yeah,
CDC.
yeah, CDC, that's it, yeah.
Yes.
But the Canadian government for, they're very.
WHO as well.
Yeah, that the rhesus monkey study that I told you, the 18 monkeys, that was actually done by Canadian scientists. So they are very actively involved in this. I mean, the usual suspects, the Americans, Canadians, and the Brits, and it's primarily driven by us.
Yes.
And is it the case that the Z-Vec that you talked about could be prophylactic as well as therapeutic use?
Yeah, well, but these drugs don't last for too long in your body, and you don't want them to. And therefore, if you are going as a healthcare worker, just like we used to do with flu, correct? You take Tamiflu as you walk into the clinic because, if you are exposed, Tamiflu would protect, but not too far in advance like you would do with, with a vaccine. Right, okay, so from that point of view, passive antibody therapy may be much better because these antibodies are going to persist much longer. The half-life of immunoglobulin is, I don't know, depending upon the class, can be 21 days. So if you take a pretty good bolus of it, you can be protected. So just like people used to take immunoglobulin when they traveled, around the world because they would give them 3 to 6 week protection, and when you come back, you are fine, So I think something like that. I mean, I thought somebody asked me about the infected individual, the gentleman. See, now there are so many survivors, correct? 50% died, the other 50% are alive, correct? And they all should be immune.
Mm-hmm.
So I don't know whether somebody is actually using those, survivors as donors and see whether we can have polyclonal serum. If the monoclonal antibodies are working so well, why can't we think of using, this purified immunoglobulin, because we have all kinds of screening, Hep C, Hep B, HIV, and all that stuff. So once you rule all of those out, I don't see why that polyclonal immunoglobulin cannot be used, as an acute, instantaneous therapy. Please.
No, they have been doing that anecdotally. In fact, for the American guy who came before he got the ZMapp, because he first offered the ZMapp to the other health worker, and one of the because he helped so many people, one of the survivors, their family got the survivor to donate blood, but then he got sicker anyway, and I think he got the ZMapp. I always get confused about that part of the story, but there are cases where people who are survivors have donated blood to people who are sick to try to give them pathways.
Yeah, but that has to be, what I'm saying, it has to be done in a regular, because you don't want a mismatch with blood transfusion, correct? So if you take collect the pooled sera, and really it has to be done as if you're developing a drug. Purify the immunoglobulin to the ultra pure and subject it to all kinds of testing to make sure we are not inadvertently transmitting, some other disease, because that would be a disaster. So I think all those things can happen once there is evidence that it is protective. That's why the Friday news about the rhesus monkeys is really, really a major breakthrough. Now, many other biopharmaceuticals have come to NIH, and NIH is asking them to work with MAP for biopharmaceuticals. Basically, they have a cDNA construct. So they want the MAP biopharmaceutical to be able to share that cDNA construct with many different companies. And not everybody is going to produce this in tobacco leaves, but some of them could be producing in mammalian cells. All you have to do is you take that cassette, you clone it into another vector, and you can put it into a mammalian cell as a factory. So those kinds of efforts, literally all of this is happening since Friday, last Friday, because that's when the rhesus monkey data came out. So it's really quite fascinating, as a scientist to follow through this story. But it's equally devastating to see so many people die the way they are dying. It's just you feel so helpless.
You're trying to put together a fundraiser to get supplies to Sierra Leone to help with some of the quarantining process.
Yes, actually, we're still in the process of developing, but there's going to be a fundraiser at the Honorary Consulate of Sierra Leone, downtown Chicago, on October 9th. So I'll talk to you and I'll get more details out to everyone.
That's fantastic. It's something we definitely want to support. And talking about boots on the ground, it's a way of really contributing where they need the help the most. And with your connections in Sierra Leone, you're going to know better than any of us in this room what the need is. Okay. Well, this has been great. I mean, I love the fact that there's basic science, there's clinicians, there's infectious disease. It's nice to see VJ Rich Doyle, who's IM, EM, now infectious disease. There's a real interest in this, and there should be. To Dr. Prabhakar's credit, he was going to lecture on the new breakthroughs in diabetes treatment, and instead he said, I have to switch. This is such a burning topic that people need to know about. So he really is, this can't get more cutting edge. We appreciate the great turnout. For those of you who didn't quite get enough food, we apologize. We didn't think that we'd be bursting at the seams. Come next month, it's the first Wednesday of every month, Dr. Steve Wine, who's extraordinary, is going to be talking about mental health in Kosovo and Tajikistan and global mental health issues. So we do this every month. We love the interest. We'll keep getting bigger auditoriums. And Dr. Prabhakar, not only is he NIH funded, over 200 publications, he's giving his heart, his time, and really busting through now in southern India and Bangalore and helping out with cardiovascular care there. A lot of his own time and vacation and dollars spent to make that happen. So he does much more than hang out in a lab looking at cleaving peptides to find the next vaccine. He's really talking about boots to the ground. He practices what he preaches. And in recognition, we want to give you a couple of things. He's active in our Center for Global Health, but I don't know if I've ever given you a coffee mug. And I know you like coffee, but you have plenty of that. Because of the effort you've put into this, we got some nice cognac for you. So if you lecture for global health, big things happen. And if you get Ebola, maybe you should just have a glass of cognac and quarantine yourself for 21 days and enjoy it while you can. So any other questions? I'm sure Prab is willing to hang out. He loves this. He breathes this topic, and there's no one at the university and no one nationally that knows more about this because of his forefront work with influenza and everything else that he's done over the years. So we're really blessed to have such an expert right here in our own backyard.
Thank you. I wish my mother was here to listen to all of this.
Well, it's all sincere. So anyone else have questions, feel free to stay. Jeannie, thank you for putting this together on the fly.
Thank you.
And Dr. Cindy, where's Cindy? She donated her computer to the cause. Yeah, make sure we get her computer back to her. Thank you.
Thank you.